We decided to show an animal cell go through the cycle. This is the first step of interphase, or G1. This orange bead here is a centrosome. It will be important later. The cell consumes nutrients. Glucose, amino acids, and some others will help the cell to grow. The cell also produces more organelles. It's also important to mention here that G0 can occur if there aren't enough nutrients or the cell is damaged. Stage 2 of interphase happens in the nucleus. This is synthesis, or S. The orange cube is helicase. Helicase unzips the DNA. Alright, let's pause it here. The green beads are single-stranded binding proteins that keep the DNA from zipping itself back together. This yellow double block is topoisomerase. This keeps the DNA from coiling up before synthesis is completed. The red blocks are called primase. Primase gives DNA polymerase a place to start. This is signified by the orange bead. The blue bead is DNA polymerase. It can only start DNA synthesis from an RNA primer's terminal hydroxyl group. This DNA polymerase here is called the leading strand. This one is called the lagging strand. As helicase unzips, the leading strand continues on, but the lagging strand has to wait for a primer to enable the continuation of its DNA polymerase. This purple block is called ligase. It seals the gaps between the lagging strand. It's important to note that later, DNA polymerase replaces the RNA primers with DNA nucleotides, and also checks to make sure that it's copied everything correctly. Synthesis produces sister chromatids. They aren't visible because they're in a relaxed chromatin form. In the future, during prophase, the chromosomes will condense and become visible. G2. It's important to note that the centrosome doubled during the S phase. In G2, the cell grows even larger. It produces some more organelles and air checks itself again. Here are some proteins. On the top, we have some regulatory proteins, and on the bottom, we have some structural ones.